Welcome to the deep dive. If you're following conservation news, you probably know about the global biodiversity crisis.
Mm.
But there's this strange paradox often at the heart of it.
hmm
While we're watching species go extinct globally, many local places, parks, forests, lakes, are actually seeing their species counts go up.
Yeah, it's a real camouflage, isn't it? It's probably the biggest one in modern ecology. You could walk into your local park, count plants, insects, whatever,
Right.
and find more species than someone did, say, 50 years ago. But the thing is, what you're often seeing are the global winners.
Ah,
The same few cosmopolitan species that are just thriving everywhere in every big city. It completely hides the quiet disappearance of the local specialists. The planet isn't just getting emptier, it's getting, well, blander.
and that process, that kind of standardization, that's what we're digging into today.
Yeah?
We're doing a deep dive into a really key paper that, frankly, forced the conservation world to get much more precise about what this homogenization process actually is, and critically, how we measure it properly.
Yeah, it was influential.
The paper is On Defining and Quantifying Biotic Homogenization, published back in 2006 by Julian D. Olden and Thomas P. Rooney.
So our mission today is basically to give you a shortcut to understanding this really important concept. We need to pull out their clear definition of biotic homogenization, BH for short,
Okay.
and then crucially walk through the scientifically sound ways to quantify it. Because like you said, the authors found a lot of people were sort of throwing the term around a bit loosely, often incorrectly.
Right. Okay, so let's start there. The definition. What exactly is biotic homogenization, according to Olden and Rooney?
Okay, so they define BH as the process where the genetic, taxonomic, or functional similarities of different regional biotas, think different forests, different lakes, actually increase over time.
Increase. Okay.
Yeah, it's fundamentally about replacement. Those unique, regionally distinct communities, they're gradually being swapped out for these widespread, cosmopolitan communities. The same stuff everywhere.
And this takes us right back to that paradox we started with.
Yeah.
We know global species diversity is down, but local diversity, what ecologists call alpha diversity,
Right, alpha.
that often seems to increase. Why is that happening again?
Well, it's usually because new non-native species establishing in an area outnumber the native species that are being lost.
Okay.
It's like, imagine a local nature preserve, maybe it loses 5 native insect species due to habitat changes or whatever.
Mm-hmm.
But then maybe 7 non-native insect species move in, species that are really good generalists. The local count, the alpha diversity, has actually gone up by 2.
Right. Looks good on paper locally.
Exactly. But the paper argues this local gain often happens, maybe usually happens, at the expense of decreased beta diversity.
Beta diversity. Okay. That's the crucial context here, isn't it?
Yeah.
Explain beta diversity for us.
Beta diversity is all about measuring the spatial turnover. How much does the species list change when you move from one place to another? It's basically a measure of uniqueness between locations.
So if you have two forests that historically had totally different sets of species,
Yeah, completely distinct.
and now they're both filled with the same common, widespread generalist species.
Then their beta diversity has plummeted. They've become more similar. That is homogenization.
Got it.
And what's really interesting here is that Olden and Rooney stress BH isn't just like a side effect of extinction or invasion. It's a distinct process driven by both, plus environmental changes. Its uniqueness comes from focusing entirely on the identity of the species involved.
Not just how many, but who they are.
Precisely, not just the raw number.
In that point, the BH is defined by species identity, not just the counts. That seems to be where Olden and Rooney really pushed the field. What was the big misconception they were tackling?
The main problem was that BH was getting used almost like a synonym for other things, like just general species diversity loss, or simply the act of species invasions, or even just changes in species richness. They argue really strongly that, no, BH must track the identity of species, and specifically the increase in similarity between different places over time. You could have invasions, you could have extinctions, without actually causing homogenization, depending on which species are involved.
Okay. How do they demonstrate that? I remember something about a simulation.
Yeah, they used a neat conceptual illustration, kind of a thought experiment. They imagined 2 pairs of butterfly communities. Let's call them Scenario 1 and Scenario 2.
Right.
In both scenarios, they set up identical rates of local extinction butterflies disappearing from one site and identical rates of non-native invasion over, say, 3 time steps. So the numbers of gains and losses are the same in both.
Okay, same turnover rates.
Exactly. But here's the twist. In scenario one, the pair of communities becomes taxonomically homogenized. They get more similar. Why? Because the specific species lost were unique local ones, and the specific species that invaded were the same generalists in both
Ah,
places.
okay. The identities mattered.
And then, in scenario two, same rates of extinction and invasion, but this time the pair of communities actually becomes less similar. They undergo taxonomic differentiation.
Wait, really? With the same rates?
Yes, because in this case, maybe different unique species were lost in each place and different new non-natal species invaded each place. So the identities changed in ways that made them less alike.
Wow. Okay. That really drives the point home. The rate of change doesn't dictate the outcome for similarity. It's all about who is coming and going.
It's completely contingent on the identity of the species. That's a fundamental shift in thinking about how we monitor change.
Yeah, it's a huge insight. And they didn't just rely on theory, right? They looked at real-world data, too. Fish and plants.
Absolutely. They backed it up with empirical data. They looked at U.S. freshwater fish communities and also terrestrial plant communities across the U.S.
And what did that real-world data show about the link between this taxonomic homogenization, the increasing similarity, and just simple species richness numbers?
Well, they found something really interesting. Taxonomic homogenization, that increase in similarity between different sites, it showed a strong negative logarithmic relationship with native species richness.
Okay, negative logarithmic. Can you translate that for us?
Yeah, basically it means that homogenization was happening everywhere, but the places that started out with really high native species richness, those places homogenize much more slowly.
So high native richness offered some kind of resistance.
Well, exactly. It acted as a kind of buffer, slowing down that standardization process. But, and this is key, it was only the native richness that did this.
Okay, so native richness mattered. What about non-native richness? Did more non-native species automatically mean more homogenization?
That's the kicker. They found virtually no relationship between taxonomic homogenization and the richness of non-native species.
Really? No relationship.
Pretty much none, which empirically proves their point. Taxonomic homogenization is a distinct process. It's not just about how many non-native species show up. It's a different, arguably more insidious aspect of biodiversity change.
That makes sense. So if BH is about identity, not just counts, it seems logical that we need to think beyond just species names.
Exactly. The authors really stress this. Defining BH properly means recognizing it happens on multiple levels or in multiple realms beyond this taxonomic one.
What are those realms?
Okay, so first there's genetic homogenization. This is down at the molecular level. It's the increase in genetic similarity between populations over time.
How does that happen?
Usually through things like hybridization between native and invading exotic species, or even just the loss of unique local genetic adaptations. Essentially, native gene pools get diluted. It's like a silent loss of future potential.
Okay, genetic. What's next?
Second, and this one has huge implications for how ecosystems actually work, is functional homogenization.
Functional, meaning the roles species play.
Precisely. It's about the jobs species do in the ecosystem, their functional traits. Functional homogenization is when the similarity of those roles increases across different communities over time. It's not about the species name, it's about what they do.
And how does that occur?
It happens when species with really unique specialized roles are lost and they get replaced by invaders or generalists that all do very similar things. You end up with high redundancy, but low overall functional diversity.
Right. I think they mentioned urbanization as an example, how city environments tend to favor generalists.
Yeah, exactly. Studies show urbanization often leads to specialist species being replaced by these non-native generalist species. The overall job pool of the ecosystem shrinks.
It's like you said earlier, swapping out unique local specialists for the same few cosmopolitan generalists.
It is. It's like, imagine replacing a complex orchestra, maybe 100 pieces,
Yeah.
every instrument vital,
Yeah.
and replacing it with, I don't know, a 4-piece rock band that just plays the same beat over and over. You still have sound, you still have species present, maybe even more species.
But the complexity, the richness of function is gone.
Utterly gone. The functional uniqueness collapses.
They gave another example, didn't they? Something about zebra mussels.
Yes, the zebra mussel, Tricina polymorpha. Classic example. When it invaded and became super abundant in the Moselle River, researchers observed a parallel, massive drop in the functional diversity of the native macroinvertebrate communities.
So the mussels came in, numbers might have stayed stable or even increased, but the variety of ecological roles just crashed.
Exactly. The whole ecological blueprint got simpler, less resilient, less interesting functionally.
Okay, so we have genetic, taxonomic, and functional homogenization. Understanding these different facets seems crucial.
It is. And recognizing the simplification of the blueprint is why getting the measurement right is so critical for conservation. If we want to actually track this, maybe even fight it, we need robust methods.
Right. Let's talk quantification. How is it typically measured, at least for the taxonomic level? You mentioned similarity indices.
Yeah, for taxonomic homogenization, or TH, it's usually quantified using indices like Jacquard's or Bray-Curtis.
What are those actually measuring?
They're basically just measuring overlap in species lists between two sites or the same site at two different times. If Site A and Site B share absolutely zero species, their similarity is zero.
Makes sense.
If they share every single species, their similarity is 1, or 100%. Homogenization tracks how that overlap, that shared identity, changes, usually increases over time between different places.
Okay. Now, the paper discusses three main ways people try to do this, and they seem quite strict about which ones are actually valid. What's the first method?
The first, and they call it the most straightforward, is simply surveying species pools in 2 time periods.
So literally going back to the same places and counting again.
Pretty much. Resurveying the exact same sites at 2 distinct points in time, crucially using comparable methods. This lets you explicitly calculate the rate of homogenization.
Any examples of this?
Yeah, they cite a study showing fish assemblage similarity increased across 62 Minnesota lakes over a 43-year period using this direct resurvey method.
That sounds like the ideal, the gold standard, but what's the catch?
Well, the big if is that the historical and contemporary sampling methods and effort must be comparable.
Ah. And how realistic is that, especially if you're looking back, say, 100 years? Historical data can be pretty patchy or collected very differently, right?
Exactly. That's the major limitation. Often, that historical data just isn't good enough or doesn't exist in a comparable format. So this gold standard might be unobtainable in many cases.
Which forces researchers towards the second method, I guess.
Right. Which leads us to method 2, which they recommend as a reasonable alternative, though it's definitely more complex. That's using extant and reconstructed historical species pools.
Okay, break that down. What does reconstructed historical mean?
It means you take the list of currently existing native species in a region and you add back any native species that are known to have been lost from that specific region, known extirpations. That gives you your estimated historical baseline.
I see. So current natives plus the ones we know disappeared locally.
Correct. Then you compare that reconstructed historical pool to the contemporary pool, which includes the remaining natives plus any established non-native species.
And this gives you a decent picture of overall homogenization, maybe linked to big events like European settlement or something.
Yeah, it can provide a reasonable assessment of the long-term impact, like Frank Rahel did for U.S. fish fauna. But there is a massive potential pitfall here.
Which is?
You absolutely must accurately account for those native species extirpations, the local losses.
Why is that so critical? What happens if you miss those? Or just assume the historical pool was only the natives we still see today?
If you fail to account for those local losses, you essentially assume the historical communities were already less unique, less diverse than they actually were. You overestimate the historical similarity between sites.
And if you overestimate historical similarity?
You will dangerously underestimate the true amount of homogenization that has occurred since then. You basically make it look like things haven't changed as much as they really have. You're assuming the system started simpler than it was.
That's a huge measurement error. Okay, really crucial point to account for those local losses, the extirpations.
Absolutely critical.
Which brings us to the third method, the one they seem to warn against quite strongly.
Yes, method 3 is basically approaches that only examine the extant species pool, just looking at what's there today.
Like what kinds of studies?
Things like comparing the ratio of endemic, unique local species versus cosmopolitan, widespread species in different cities now, or looking at how species similarity changes along an urban to rural gradient today.
Why is that problematic for measuring homogenization?
Because without knowing the historical degree of similarity, how different or similar those places were before the changes happened, you simply cannot assess whether or by how much they have truly converged over time.
You're missing the baseline.
Completely. You can describe patterns today, but you can't actually prove homogenization, the process of becoming more similar over time, without establishing how different they used to be.
Got it. So avoid methods that only look at the present day if you want to measure actual homogenization.
That's their strong recommendation.
Okay, so we started this deep dive noting this paradox of local gains versus global losses,
Hmm.
and how homogenization focuses on the loss of uniqueness. Bringing it all together, what's the biggest takeaway from Olden and Rooney?
I think the main synthesis is that for conservation biology to move forward effectively, we have to get past this idea that biotic homogenization is just another way of saying species richness change or invasion impact.
It's distinct.
It's distinct. And if we fail to appreciate the nuanced, multifaceted nature of it, genetic, functional, taxonomic politicality, we risk completely overlooking really significant ecological and evolutionary consequences. Consequences that can happen even when local species counts look stable or even deceptively healthy.
And the paper finished with some pretty clear calls to action for researchers, didn't it? Like adopting these standards.
Yes, very clear. First, explicitly recognize those three realms: genetic, taxonomic, functional. Don't just focus on species names. Second, prioritize tracking the identity of species over space and time, not just counting numbers. Who is where and how is that changing?
Right, the who, not just the how many.
And finally, they implore researchers to use rigorous, scientifically defensible methods for quantification. That means, ideally, the two-period surveying method.
The gold standard, if possible.
Or if not, that carefully reconstructed historical baseline method, making absolutely sure to account for native species extirpations.
So if this process really is simplifying the planet's biological blueprint, as you put it earlier, the stakes feel incredibly high.
They really are. I think the ultimate implication is that we need to do a better job educating everyone, policymakers, the public, about this specific component of the biodiversity crisis.
Because it's not just extinction numbers.
Exactly. If we keep looking at biodiversity loss only through the lens of species richness decline, we risk putting our conservation efforts in the wrong places. We might be fighting to keep local counts high while inadvertently allowing the unique character of the world's ecosystems to just fade away.
Losing distinctiveness itself.
We're not just losing species, we're losing the planet's variety, the unique biological signatures forged over millennia. And that might be the most profound loss of all.